All right, everybody. Thanks so much for coming out tonight. We have a really special venue. So maybe first off, huge thanks to Pillar VC for hosting us tonight.
Thank you guys for coming. As I said in the email, I'm John. I'm a venture partner here at Pillar. Pillar drives founder-led bio. So our goal is to support and encourage people in building their own companies. And then we also, as part of that, do a ton of community events around all sides of both biotech and non-biotech entrepreneurship and startups and science and R&D. I've known Chris for years, and I've been deeply involved and interested in peptides and protein drugs for longer than that. So when I started coming to these BPTMC meetings, suddenly struck me that there might be an opportunity to split the difference between MIT and HMS and host an event here at Pillar. So took a little while, but I'm glad we got it all figured out and glad to see such a good audience. So hope you guys enjoy. And I'll be around afterwards if anyone wants to chat. But thanks again for coming. Appreciate it.
Fantastic. Thanks again. Also, thanks to our gold and individual contributor sponsors, Twist and Nexomics. Really appreciate the support. For any folks in industry out there who would like to see your logo on the screen here and in the acknowledgements, it's very achievable. So just come chat with me. We can make sure that your company gets recognized. So yeah, without further ado, really excited to introduce our speaker tonight, Andrew. So Andrew is at MIT and importantly has a K99 and is on the faculty job market this year. So really excited to see your talk tonight. And yeah, please take it away.
So thank you so much for that introduction, Chris. And thanks, John, for this great venue. It's great. And thanks also to Sergey and Nick for chance to present here today. So yes, as Chris said, my name's Andrew Savinov. I'm a postdoc with Jinwei Li at MIT currently, and also co-mentored by Stan Fields at the University of Washington. And today I'm really excited to share with you my postdoctoral work on the massively parallel discovery of peptides to inhibit protein interactions in cells. So in my research, I'm broadly very interested in the relationships between sequence, structure, and function across scales in biology. And by this, I mean both how sequences of biopolymers like proteins and RNA fold into specific structures, which impart particular biochemical functions, but also then how those functions are propagated to the cellular level, ultimately driving differences in fitness under different physiological conditions. And that, of course, includes the roles of every single residue in these polymers in their function. So I'll first briefly tell you about some of my graduate work, just in a brief overview, where I first got into these questions, thinking about the RNA sequence structure function relationship. And this is when I was a PhD student with Steve Block at Stanford. And in that work, I looked at both the relationships between folding and catalysis in RNA and the roles of every residue in this model RNA we studied in catalytic activity. And then I'll spend the remainder of the time, and the most of our time today, talking about the work I've done now in my postdoc, looking at protein-protein interactions in living cells. So in my graduate work, I was really excited to delve into this molecular sequence structure function relationship of RNA. So I studied this model ribozyme. It's a self-cleaving RNA that also is actually a genetic switch, which controls bacterial cell wall synthesis. So I used two complementary approaches to look at this system, and one was force spectroscopy with optical tweezers, where we could actually capture single molecules of this RNA between two polystyrene beads, which we trap with high-powered focused lasers, and we can then measure both the distances of the system as it folds and unfolds, and also the forces involved to get at the entire energy landscape of folding, right? And we also used massively parallel biochemistry, which I'll tell you about in a second, to study the sequence function relationship. So in terms of the single molecule side of that work, we basically used this approach that I just told you about to reconstruct the full folding free energy landscape of this RNA from the fully unfolded single-stranded RNA to the functional ribozyme. And what we saw was that we actually had this key folding interaction that I'm highlighting, right? Where the last step in folding is actually the formation of just a single duplex. And this duplex is actually only marginally stable. So it's only about 1 kcal per mole, right? Or 1.5 kT. And interestingly, in contrast to the dominant model for how ligand-dependent RNA switches work at the time, the binding of a saturating amount of the ligand didn't change the stability at all. And so there's this really marginally stable interaction that controls function in this system. And it's, of course, interesting to think about how much that kind of concept generalizes. I think we're seeing more examples over time. And we also were actually able to do single molecule enzymology of the RNAs trapped in this approach, right? And so we could actually measure the dependence on force of both the cleavage or catalytic activity of the ribozyme and its folding to understand the relationship between the folding and catalytic activity. And what we basically found is that this single fragile folding interaction totally controls the ability of the ribozyme to perform its catalytic activity. And so we found that this is a system where actually this key RNA switch in bacteria is basically controlled by just a single marginally stable interaction that's not stabilized by ligand binding. And it's essentially just a stochastic process that actually underlies a lot of important functional features in this organism. Now, we also wanted to understand the sequence function landscape, right? And this is a question where single molecule methods generally don't scale so well. So we wanted to really be able to look at the whole mutational landscape, or at least a nice portion of it, and in particular, all the single and double mutants of the ribozyme. So what we did is we worked in collaboration with Will Greenleaf's lab at Stanford, and we built what is called an RNA array, where you can actually transcribe clusters of DNA directly on a sequencing chip, say an Illumina sequencing chip, into clusters of RNA. And so we had this library of transcription templates. We made libraries of ribozymes, including all possible single and double mutants. And we got data that looks like this, where we have the cleavage rate effects of all possible single and double mutants, right? So there's a lot there, and there's a lot that we did with these data. But in terms of just the brief overview today, we found that one way to analyze this that was very informative was to look at rescue effects, or epistasis, where we found that we could look at every double mutant and its catalytic activity compared to the single mutants that comprise it. And that let us actually, for example, reconstruct the entire secondary structure of the ribozyme purely from measurements of function, right? And we could also see a lot of other features on this map besides those diagonal features. And essentially what we saw is that we can reconstruct the secondary structure, but we can also actually see the catalytic relevance of each tertiary contact to discover novel tertiary interactions that can form as you move along the mutational landscape, right? And excitingly, we saw that actually there's a lot of plasticity in the secondary structure that still allows for function. And we can sort of map that out as we looked at all the double mutants. Okay, so that wraps up what I wanted to tell you about regarding my PhD work. And then going into my postdoc, I was really excited to continue to think about sequence structure function relationships, but now in the context of the living cell. And in particular, I was really interested in the consequences of these molecular biophysics in their natural environment, right? In this really crowded and complex cellular environment. And one really universal manifestation of that question is that of how biological function is really intimately dependent on the interactions between proteins, right? Both how proteins fold, of course, but also how multiple proteins come together into higher order complexes. And these higher order complexes, in turn, whether they be transient or stable, really underlie a lot of the higher order functions that we think of as characteristic of cellular life, right? And one example that I'll come back to today is we can think about the cytoskeletal protein FtsZ, which needs to form these filaments that then form the Z ring required for bacterial cell division. So of course, we're very motivated to understand how these structural interactions of proteins play out in cells. And a great tool for understanding protein interactions is perturbing, right? And a fantastic tool for doing that is provided by peptides. Peptides are capable of binding to larger proteins at specific target sites, as many of you here are very well aware, and they can then modulate the structures, functions, and interactions of those proteins. And it's becoming increasingly clear that this is actually something biology figured out a long time ago, and organisms have naturally evolved a vast array of what are variously known as mini proteins, small proteins, micropeptides, and so forth, which are basically biologically encoded peptides that have important functions. And frequently, these mini proteins actually regulate the functions of larger proteins in response to various stress conditions. And in fact, the extent of this phenomenon is so large, as we're becoming aware of this, that people have started to call this sort of dark matter of the protein. As has received some attention in the news recently, it's also possible to design, of course, peptides to bind specific targets. And the ability to do that has, of course, just taken off more and more since this particular single example I'm showing you here. And so we can, of course, build peptides to bind specific targets. And of course, it's a very promising avenue for targeted therapeutics. And so I've become very interested in one specific class of peptides, which are fragments of full-length protein sequences. So protein fragments inherently have some capability to reproduce native or native-like interactions of their parental protein. And so they potentially can actually compete with their parental full-length protein for its native interactions in cells. And if they can do that, they, of course, can potentially then act as a dominant negative inhibitor, where they can bind to a site but lack other features of the full-length protein. And fragments like this aren't just something we can think about synthetically. They actually also arise in cells naturally through multiple pathways, including errors in gene expression. And you might imagine then, based on what I'm telling you, that fragments like this would also pose quite a threat to the health of the cell. And actually, there's some evidence that that's the case, because there's also a lot of quality control processes that suppress the formation of protein fragments. Now, historically, we've developed a lot of great knowledge on what truncated proteins can do, sort of protein fragments largely based on studies of naturally occurring truncations due to mutations at the DNA level, as well as splice variants. And I really wanted to develop a systematic understanding of what small protein fragments are capable of and what we can learn from them. And that would really be enabled by a high-throughput approach to measure the inhibitory function of these protein fragments, both across any given protein, but also across many proteins. So today I'll be telling you about my work in which we actually developed an approach to do just that and to directly measure these effects in living cells. And then I'll also tell you about how we were able to leverage this approach to really determine principles of fragment-based inhibition and show that these kinds of protein fragments are actually generalizable genetically encodable inhibitors. And finally, I'll tell you about these high-throughput computational approaches we've now developed to be able to predict these inhibitory protein fragments at the same scale that we can measure them experimentally. So I'll first tell you about this experimental approach we've developed. So I'll describe the massively parallel experimental assay and then show you how this approach works using the example of FtsZ. So in this approach, we build a library of DNA oligos encoding fragments of the proteins we're interested in. And in this case, we built a tiling library of fragments tiling across the proteins of interest with the 1 amino acid step size. We then clone these fragments into an expression vector, and we transform them into cells. And in this case, that was E. coli. And we can then grow the cells in a massively parallel competitive growth assay under conditions in which the parental full-length proteins that these fragments are derived from are important for growth. So fragments that inhibit their parental interactions will inhibit growth, and we should see the frequencies of cells carrying those fragments decline in the population over time. We can quantify that through high-throughput DNA sequencing, so we can really quantitatively measure the frequencies of each fragment in the population throughout the selection. And we can then map back inhibitory and neutral fragments back onto the sequences of their full-length protein. And you can imagine that this would then let us actually build a map of inhibitory activity as a function of fragment sequence position, as I notionally show here. And so I'll use the example of FtsZ to show you how this works and what we can learn with this approach. And the reason this is a great example for us is because we have well-defined and well-characterized structural interactions that are then responsible for this protein's very direct role in cell fitness, the ability for the cells to divide normally. So here's what the data actually look like, going from that sort of notional cartoon. We have the inhibitory effects along the vertical axis, where this is what we're calling enrichment, which is the log of the relative frequencies, where larger negative values here correspond to greater inhibitory activity, as a function of the sequence position of the fragment. So every point here is a single 30 amino acid fragment of FtsZ tiling across the protein with the 1 amino acid step size. So we see that there indeed are very characteristic peaks of inhibitory activity at specific sequence positions. So the question, of course, is then what are these data telling us? Can we make any sense of this, right? So to do that, let's take a closer look at the structure of FtsZ. So FtsZ forms these filaments that I mentioned. And if we take a closer look at a given monomer from the filament, where these monomers come together in a head-to-tail fashion, we can look at it and we can see, it's recolored now from the end to the C-terminus. And there are basically 3 approximately reciprocal portions of the interface we can define, which I'm calling here just sites 1, 2, and 3 on one face of each monomer, and 1', 2', and 3' on the other face. And so if we then map these structural features of FtsZ onto our data, we can see what that gives us. And it turns out that, for example, the strongest central peaks, interaction site 1 here, indeed maps to interaction site 1, right? So we've colored that corresponding region on the protein and indicated it overlaid on our data. And we also saw, similarly, that the reciprocal interaction site from the other side of the filament, site 1', also gives us an inhibitory peak. And we see the same thing for interaction sites 2 and 2'. So we're actually mapping out a good portion here of the interface for filament formation with our inhibitory fragments. So that was exciting to see. And you might imagine then that indeed these fragments are actually inhibiting filament polymerization. And that's why we see this effect. And I'm just showing you here another view of how these really map out a good portion of the interface. And so we were able to confirm that through a light scattering assay you can perform for filament polymerization. We can show that in vitro, these fragments indeed can inhibit filament formation. Now, you might notice that we don't see any inhibitory peaks for interactions like 3 and 3', which are comprise the remainder of the interface of filament formation. And this actually turned out to make sense as well, because Shoemaker and colleagues who had done the crystallography of this complex actually showed that this particular region of the interface has a high B factor, and so it's thought to be flexible and not contribute much to the binding energy. So perhaps fragments just bind very poorly there and don't really affect the interface. So overall, from what I've shown you, we can see that we can actually identify specific protein fragments that inhibit protein interactions, and we can actually make measurements of this effect directly in cells. So then I'll show you next how we were able to actually learn principles of fragment-based inhibition across diverse proteins and really show that these protein fragments are generalizable and genetically encodable inhibitors. So I'll show you how the results generalize beyond FtsZ, how we can actually explain how well this approach works for fragments from different proteins where we see differences in efficacy, and connect this all actually to what we see with naturally evolved mini proteins, which I mentioned previously. Finally, I'll show you how we've actually delved into the roles of fragment length and other simple biophysical properties, and to what extent they can explain what we have here, and whether we can actually get to design principles. Okay, so because this is a massively parallel method, we could really look at a very diverse set of proteins. And we specifically selected proteins with very different structural and functional features to empower us to learn some general principles of fragment-based inhibition. And so you might see one of your favorite proteins here, and we can talk about it more, let me know. But I'm just showing you a few examples here. And what we see in general, just as in the case of FtsZ, is that we see these inhibitory protein fragments mapping to key interaction sites of these proteins. So for example, in the case of RPLL, which is in the upper left-hand corner here, this is a ribosomal subunit protein, and you can see that there's this very strong inhibitory peak mapping to the interface for forming part of the ribosome stalk. So overall, what we find across these different proteins with diverse structural and functional features is that protein fragments readily inhibit protein interactions in vivo. And the functional fragments at this expression level are actually really widespread, where about 25% of the peptides tied across the proteins we look at have a significant effect. And so we're really excited now about the potential of these protein fragments as novel genetically encodable inhibitors towards developing new peptide drugs. So another thing we looked into is whether, yes, please go ahead, Chris.
It's really exciting. I'm just wondering, what sort of affinities do you think these peptides are having for the target protein?
Yeah, that's a great question. So we would love to do a direct measurement of that and to do that systematically. We don't have that yet. I'll show you later. We've done Rosetta modeling of the protein fragment binding modes that we model, comparing that to the Rosetta energies for known structurally characterized peptide protein binders. And so we do see that they are pretty comparable to similarly sized small peptides in the predicted binding energy, but we don't have a direct measurement. We also see that when we did the in vitro measurements of filament polymerization, a 4:1 ratio of peptide to protein, which is really not that much, you have a 20% reduction in filament polymerization. So we haven't turned that into a KD at this point, or a KI, but it's certainly, at least some of them are capable of a decent affinity to achieve an effect like that. Yeah, please.
Since we're doing questions right now. So if I understand correctly, your the way you're measuring your enrichment is due to growth, right? Growth of the organism.
Yep.
So I guess I'm wondering, how do you, you did the in vitro thing, you sort of, okay, it affects polymerization of this one protein, but how do you really know that you're not just making a peptide that's amphipathic and it's just going to pop the cell membrane or something from the inside? How can you really know that the effects of inhibition are due to activity on the protein of interest as opposed to something else, or just popping the cell?
Yeah, great question. And we'll be getting into different facets of that as we go along, but briefly.
Also, maybe repeat the question briefly. So
Yes, so the question is, since our assay readout is based on effects on cell growth, how do we know really that the fragments are actually acting through this mechanism of inhibiting these interactions in cells? Right. And so, yeah, we'll be touching on that through various approaches. But basically, one thing we see, as I'll show you actually on the next slide, is that these effects look exactly like what you'd expect in general for titration of the native interactions based on their concentration dependence. So we'll give the game away for that one. And we also, which you really wouldn't expect if it was acting by some other mechanism. I think what you're saying in general is absolutely happening at some frequency, right? So I think on average, we see a strong driving force from these fragments reproducing native-like interactions. But I think on top of that, there's a lot of novel functions that we're actually, in some sense, are equally or even more exciting, right? So thinking about new biology, new kinds of functions you can see here. And my guess is that based on the number of peaks we could actually map to a known interface, there are probably half or so of the peaks we see that we don't have a good explanation for based on interactions we currently know about. So that suggests that maybe, say, half are driven by these native-like interactions and half are not. And I think one way to think about it is that if a fragment can reproduce a native-like interaction, it's actually evolved to form that interaction, right? And so maybe it's not so surprising that a small peptide can, if it can fold well enough to bind or bind upon folding, it's actually pretty likely to act by that mechanism if it's able to bind, because that's sort of what's evolved to bind the best, right? But I think there's definitely going to be cases of just absolutely novel functions as well, right? Yeah, we'll come back to other aspects. We can talk more. Yeah. Yeah, please.
Do you know, do these peptides have any conformation by themselves, or they kind of mold on your polymer, and that is the way they stop the polymerization?
Yeah, in the case of FtsZ, right. Yeah, so the question is, do we know if these peptides have any real conformation in isolation, as in before binding, right? Or if they're basically folding upon binding largely. I think they're largely I'm guessing they're largely folding on binding, and we have some deep mutational scanning data I'll show you later, where the signatures that we see are consistent with that idea based on how sensitive many sites are in terms of the folding in particular. So I think absolutely there's a lot of folding upon binding happening. And one way to make better versions of these inhibitors would be to make them fold better, even in isolation, right? Yeah. But yeah, and so I'll show you some examples of some data we have on that later through the DMS. Yeah.
You might have said this, but how do I ions?
Yes. What I've shown you so far is all 30 amino acid fragments. And then we've actually also looked at length dependence, which I'll get into briefly. And there's some interesting trade-offs there. Yeah. Yes, please go ahead.
No question. So you said about half of those might be conformational as you expect, and the other half is, let's call it a phenotypic screen. Right? Did you check in unstructured protein world? Did you find any of these hits?
Yeah, so the question is, if about half of fragments are sort of, we think, acting as a native-like inhibitor, and then maybe half are doing something else, have we looked, for example, in disordered proteins for if we see anything interesting there? And yes, so we actually do have some examples of that I'll get into a bit later. I won't spoil the surprise, but yes, we have. Yes. Okay, great. So if there are no further questions now, I'll move on for now. Great. So we saw that although this generally worked really well, there were actually quantitative differences, and sometimes to the point where it was a qualitative difference in one case, in how well this actually worked on different proteins. And two really striking examples on two ends of the spectrum were with GROES, we saw almost no inhibitory activity. And then with fragments of DNA gyrase subunit A, we saw extremely strong inhibitory activity for these peaks. So we wanted to understand what would drive these differences, right? And of course, there's so many possibilities because this is in living cells, right? And these proteins are very different. So this could be differences in localization, structural features, the number and types of interactions these proteins form, and so forth, right? And not to mention interaction strength. But we actually found that a single simple feature, I think I have to click again, simple feature can actually explain these differences in susceptibility for almost every protein we looked at. And that's just that the average inhibitory effect of the fragments tiling across a given protein had this very strong correlation, we can see it linearly here, right, with the, in this case, the log of the full-length parental protein. So the fragments are all being driven at the same expression level, right? But of course, the parental proteins, we're not perturbing in any way their expression, so they're all at their normal levels. And what we see is that there's this very strong linear correlation where the more of the full-length protein you have, the weaker the inhibitory activity is, which is actually exactly what you expect for titration of these native interactions being a major driver of what we're seeing, going back to your question. And so we were actually pretty excited and surprised at how strong that correlation was. We do see an exception here, an outlier, which is fragments from this ribosomal protein that I mentioned, L7, L12, or RPLL, are much more inhibitory than you expect from the high concentration of the ribosome and the ribosomal proteins. And we'll come back to that later. There's a number of reasons we think this might be happening. One is that with a growth assay, there's an outsized effect of anything you do, we think, with ribosomes. And we also think ribosome assembly is complex and multi-step. So there may be kinetic effects where you don't have enough protein to titrate things away, but you might have enough protein to really gunk up the works in ribosome assembly and function. And there's some other tidbits we'll get into later. So there are some interesting exceptions. And in some ways, of course, the exceptions are some of the most exciting things to follow up on. But overall, we see that this really clear signature that titration of the native protein interactions is really the major driver of the fragment-based inhibition we see in cells. And we also saw that these protein fragments actually recapitulate naturally evolved mechanisms. And so, for example, if we look at the interface that we map out with our inhibitory fragments of FtsZ, it actually perfectly matches the interface targeted by a mini protein of just about the same size, which is MCIZ. So this is a B subtilis protein that actually regulates and turns off cell division during sporulation. So it was exciting to see that we can kind of converge on the same mechanisms that nature has already evolved. We also see that there are these very specific transmembrane fragments from this membrane-spanning protein LPTG, which is in the inner membrane in bacteria. And these specific alpha helical regions are actually inhibitory. And this is actually very reminiscent of a number of natural mini proteins, which are basically just single hydrophobic alpha helices, go into the membrane and regulate various proteins and protein complexes, such as this drug efflux pump. Now, I promised we'd talk about fragment length. And so we looked for a number of proteins at a few different fragment lengths, 20, 30, and 50 amino acids. In the case of the protein I just showed you, we went down even to 14 amino acids. And we were curious how fragment length affects function, right? So what we saw at first, if we just look at the average inhibitory activity across all fragments, is that longer is just better, right? Longer fragments give stronger inhibitory effects in general across these different proteins. But actually, when we looked at a metric for the site-specific inhibition, we saw that there was an optimum somewhere in between 20 and 50 amino acids. And so it seems like it's not always better to just use a longer fragment. So you might wonder what's going on. And so if we use, again, FtsZ as an example, what we see is that as you go from 20 to 30 to 50 amino acids, the average inhibitory activity increases, but actually, once you get to 50 amino acids, you're seeing a lot of non-sequence specific inhibition. The whole baseline shifts down. So we think these longer fragments start to have a lot more non-specific effects, though you can still see the signatures of these specific inhibitory activities, right? And we think this makes sense because the actual interfaces that we're talking about here are quite small, right? Say 5 to 10 amino acids. And then you presumably need enough additional sequence to sort of give a structural scaffold for that. But at some point, adding more amino acids may not be a net good because you could start to have all sorts of alternative binding modes to other components of the cell. So we think, and you can really see this because actually the peak of inhibitory activity, it's not just that the baseline shifts down, but the peak of inhibitory activity with the 50 amino acid fragments is weaker than with the 30 amino acid fragments. So we think in some sense, these longer fragments might start to compete with themselves for their most productive interactions, which we think are generally, on average, the native-like interactions when it's some key protein interaction for cell division, say, or for cell growth. Now, I don't want you to get too upset about 50 amino acid fragments in that they still have this really clear hallmark for the titration of native interactions. So although we see there's more noise effectively, right, that these fragments have evidence that these fragments can probably do other things to a larger degree, we still see this very clear linear correlation with the 50 amino acid versions of the fragments. All right, so we also wanted to see to what extent simple, yes, please, go ahead. I have a question.
Just curious if you happen to have done an experiment for one of the lowly expressed ones, but you express a really strong inhibitory fragment, and also co-express strongly the protein that in the native cell is expressed weakly, that can be rested back up.
Yeah, that's a great question. That's on the list. We haven't done that currently, but yes, it would be fantastic to kind of map out the sort of genetics, right, by looking at overexpressing different proteins and sort of modulate this titration from both sides. What we're doing right now in some of our newer work is we actually have a titratable version of this library where we actually can have a calibrated expression level. And so from the fragment side, we're going to have a lot of power yet to figure out how strong the effects are and how much they depend on concentration. Yeah. Yes, please.
So given the fact that there's an optimal lattice of your inhibitor, I was wondering, do you think the optimal inhibitor should have a higher binding affinity than the naive protein, or lower or similar?
Great question. Yeah, I mean, higher affinity is probably just going to be better. And as I'll show you later, now we've started mutagenizing these fragments, and we can make them better. We can make them better than the native fragments. And so that's exactly the direction we're going. I think for many applications, you just want a stronger binder that basically, but you want to make sure it's still doing the same thing. Right. So we wanted to see to works that simple biophysical properties could explain what we're seeing here, and if we could maybe just start to design fragments with those properties, right? And so some properties you might imagine are driving a lot of this, and this is actually something that came up earlier in some of the questions, is what about hydrophobicity, right? Are these just like largely hydrophobic fragments that form pores? That would be a kind of boring mechanism that could be happening. What about charged, highly charged fragments? And what about stability? Like, are we seeing a lot of effects of peptide stability? And of course, are there secondary structure features driving what we're seeing? So we looked at a number of these simple properties, and we did an ANOVA analysis. And basically what we see is that none of these really simple properties really have much predictive power. The two things that have the most predictive power that I'm showing you here are actually things we already talked about, which is the fragment length, right? And the protein of origin, which of course is due to the concentration dependence effect that we talked about. But charge, hydrophobicity all had basically completely negligible effects in terms of predicting which fragment would be a good inhibitor. And so this is really consistent with something that I think you all are very familiar with, which is that we really think the effects of these fragments are largely driven by unique properties of the specific interaction interfaces they're targeting. And so to understand further how to make a good fragment, we need to think about understanding the properties that make for a good binder at a good interface, right? And briefly, just to show you another view of what I was just saying, right? We see these very clear trends driven by fragment length and by the protein of origin on average. But this is just to show you, this is showing all the fragments at each length and all the fragments for each protein, that there's a lot of spread. And we really want to understand the variation in inhibitory activity for every specific fragment, what makes a good inhibitor. And to do that, we think we'll really have to understand the interfaces and their detailed properties. Okay, so to recap this section, I showed you that inhibitory fragments generally map to key interaction sites of proteins, and that inhibition is really driven largely by this effective titration of native interactions in living cells. And so we think these protein fragments really are generalizable inhibitors, which we're excited about. Now, I also showed you that there are functional trade-offs with fragment length, and we can talk more about some of the reasons you might actually sometimes prefer a shorter fragment, even if it gives weaker inhibition. And I also showed you how the evidence suggests that there are really more highly specific properties of the interaction that we really have to think about if we want to start designing these fragments from first principles. Okay, so now I'll tell you about the computational approaches we developed to really predict these inhibitory fragments. And so I'll explain our method for computational fragment scanning, which we've called FragFold, show you how this approach is effective in predicting both known protein-protein interaction inhibitory fragments and also novel ones, including novel, previously unknown protein-protein interactions. And bringing it all together, we'll then get into how we actually have now done deep mutational scanning experiments, as I mentioned previously, in order to both get at experimentally the roles of each residue in function, but also how that then maps onto the predicted binding modes and the folding modes from our models. And so in this part of the work, I basically really wanted to predict which fragments are inhibitory, and I wanted to really do that by mirroring as closely as possible the high-throughput experimental measurements in a computational way at the same scale. And so this is the point at which I teamed up with Savinov Swanson, who's here. Thanks for coming, Sebastian. And he, at that time, was a grad student with Amy Keating at MIT. Now he's a research scientist at Novo Nordisk. So we reasoned that AlphaFold predictions should provide a really great way to do this, because we could, in parallel, both screen for inhibitory peptides that are predicted to bind to our target of interest, and also provide support for mechanistic interpretation of the high-throughput data that we generate. So we developed this approach that we call FragFold. And the way it works is that we basically computationally fragment the proteins of interest. And in this case, we did it exactly as in the experiment, with 30 amino acid fragments tiling across each protein with the 1 amino acid step size. And we then did massively parallel modeling using AlphaFold of the predicted binding between each fragment and a set of relevant protein binding targets or suspected binding partners. And importantly, we did not use AlphaFold multimer, which was trained using full-length protein-protein complexes to do this. Rather, we actually used the original monomer weights of AlphaFold2 because we didn't want memorization from the PDB of known full-length protein-protein interactions to be driving the predictions of how a fragment can bind to a protein. And we also, as we'll get into briefly further, didn't do any pairing of the MSAs, these predictions. So we did these parallel predictions, and we then used metrics we developed that I'll tell you about in a second for the extent of predicted binding in the structural models produced by AlphaFold to sort of score each model as a function of its tiling fragment position across the full protein sequence. And we could then directly compare the predicted binding from FragFold to the measured in vivo inhibitory effects of the fragments in cells. And in terms of further details on how we do this, I'll just mention that we are using the AlphaFold implementation. So thank you so much, Sergey, and other folks who worked on that. And we build the MSAs with MMCX2. We then do a few tricks, right, that others have also landed on. One trick is that we pre-generate all the MSAs so that we're not calculating MSAs every time we run a prediction. Very important for what we're doing here. And we actually calculate the MSA for the full protein that we're fragmenting first, and then chop up the MSA to get fragment MSAs. And then we put together the MSAs as input for AlphaFold in a completely unpaired way, just with row-wise concatenation. And we feed that into AlphaFold. All right, so what do we get? Here I'm showing you again the inhibitory activity from our experimental measurements, now in green, for fragments tiling across FtsZ. I've just flipped the axis so that larger values of inhibitory activity are now positive. And I'm overlaying that with the FragFold-predicted extent of binding in blue, where I'm using a metric we developed, which we're calling weighted contacts, which is basically the number of contacts within a reasonable distance from the models produced by AlphaFold, multiplied by the IPTM confidence metric. So what we see is that it was actually quite exciting to see, right? In green, we have the inhibitory peaks. For every inhibitory peak in green, we see a corresponding predicted binding peak in blue. You will notice that there's also one predicted binding peak in blue that stands out as having no corresponding inhibitory peak. And that actually turns out to map to this region 3 and 3' of the interface, where I mentioned to you that there's a lot of flexibility. And so it seems like binding is actually weak, and perhaps FragFold or AlphaFold can't really tell that apart because it can't really do anything about entropy, right? And so, but we were very excited to see that for every inhibitory peak, we did get a predicted binding peak. And not only that, and we'll come back to that in a second, but we also saw, we'll go back in a second, we also saw that for every one of those predicted binding peaks, the predicted binding mode was native-like. But notably, it's not identical to the native binding. And I'll remind you again, we're not using multimer here, right? So you see that you're predicting binding, but it's actually the subtle differences that come up are interesting. And as we'll see, they actually agree better with our experimental data in many cases than the model from the full-length protein. So going back a second, many of you may be familiar with various confidence metrics people have used to score models from different implementations of AlphaFold interaction prediction. And we came up with this metric of the predicted binding from number of contacts multiplied by confidence, right, to give us this weighted contacts, which worked really well. But we actually also tried metrics others have used like IPTM and PLDDT. And it was really striking to what extent those failed to predict these inhibitory fragments. So at least when you're using AlphaFold 2 with the monomer weights, you really can't use those approaches to get good prediction of actual inhibitors that agree with experimental data. All right, and so what I showed you here for this example fragment held up across all of the different peaks along the interface for FtsZ. And so of course we wanted to see how this generalized to other proteins. And what we saw is that just in the case of FtsZ, across all of our known inhibitory peaks that map to known protein-protein interactions, we had about 87% of those mapping to predicted native-like binders from Frog. So that was great to see. You might be wondering about what the other 13%. And what we actually saw in a couple of cases was really interesting, which is that we saw failures where the RMSD was quite good. So the fragment was almost there, but it wasn't close enough for physical contacts, right? And so on the right, I have an example of a successful prediction. On the left, an example of a failed prediction from this data set. And I think it's a good reminder, that AlphaFold isn't really a physical model per se, and you can predict something that looks like binding without things actually touching each other, right? And we're actually working on some further developments to see if we can sort of coax these kinds of close near misses into actually predicting binding. So we wanted to go further, right? So everything I showed you so far was looking at known protein-protein interaction inhibitory protein fragments where we already had the experimental data first, and we knew this mapped to a known interaction interface. We wanted to see if we could actually predict all of the inhibitory peaks, right? See what fraction of the inhibitory peaks could be predicted, and similarly see what fraction of all of the predicted binding peaks were actually inhibitory, right? This is especially important if you want to start from the predictions and design a peptide inhibitor based on that. And so to do that, we calculated these fractions for all of the peaks, not just peaks where we had some model going in. And we also did this in a way that was purely based on peak overlap, rather than trying to think about structural features going in. And I'll also say that we developed automated approaches for peak calling in both the experimental data and the predictions from FragFold in order to, again, get away from bias in what we're calling a peak. Right. So overall, we saw that 64% of inhibitory peaks were predicted across all these proteins. So it's lower than that 87%. This makes sense. Not every kind of protein-protein interaction can be predicted by AlphaFold, or certainly not equally well. And there also could be interactions with other things like RNA, and we see that in some of these structures. So it makes sense that it's a lower value, but it was a good number to see nonetheless. We also saw that about 68% of the predicted binding peaks or clusters were inhibitory. So this latter number is particularly important, as I mentioned, if you want to really predict binders and then try to use that information with no experimental data going in. So overall, we think based on these results, FragFold can be automatically applied to any protein you're interested in. And we think this high success rate really opens up proteome-wide applications that we're excited to be pursuing. So given the success we saw with FragFold, we wanted to see if we could predict novel inhibitory binding modes, novel interaction modes between proteins that have no existing experimental data. And so to do that, we actually looked at an intrinsically disordered protein, or rather an intrinsically disordered region of a protein. And this was the intrinsically disordered C-terminal tail of FtsZ. And so this is a region of the protein that forms a number of really important interactions in cell division with other proteins. We know about these interactions, but we don't know about their structural features or what the interface is. And we also actually saw, which was nice, that there was an inhibitory peak in that same region. So we know experimentally there are effects of fragments in this region, but we don't know what, if anything, those map to in terms of predicted binding. So to look into that, we did the FragFold predictions. And excitingly, we saw that for every one of these examples, FragFold actually predicted novel, previously unknown binding modes that agreed with really orthogonal genetic and biochemical evidence for what these interactions look like. Because people have done a lot of nice genetics and biochemistry, as I'll show you a few examples. So for instance, one of the proteins that the FtsZ C-terminal tail binds to is known to be FtsZ, but we didn't know how, right? But we did know biochemically that it inhibits the GTPase activity of FtsZ, and so it actually acts as an autoregulator of the protein. And so the FragFold model, right, actually predicts that it binds directly between the monomers in the filament right at the GTPase active site. And similarly, for the case of FtsZ binding to MinC, there's been a lot of nice work from Joe Lukenhaus and others, where they showed that there are specific residues that when they're mutated really abrogate this interaction. And we saw those exact residues from both sides being involved in the predicted interaction. And we saw similar results for ZipA and for FtsA binding by these fragments. So overall, these results suggest that AlphaFold really has learned some general principles of protein peptide binding. And of course, there's increasing convergence about that from many groups and a lot of work that's been going on. And this really, I think, supports broad applications of FragFold to interpret experimental data. One thing we saw that was really enticing to us was that we wondered, well, why don't we ever see these interactions? Do we ever see these interactions if we just look at full-length proteins, say looking at the C-terminal intrinsically disordered tail? And the answer was no. So this is an example where modeling protein fragments lets you predict novel interactions and interfaces that you can't predict successfully when you model with the full-length proteins. And we saw that very clearly where we can, with FragFold, predict the peptide binding of the GTPase active site. But if you just look at a single full-length monomer of FtsZ, it doesn't predict this interaction. And neither do you get that prediction if you look at a dimer of FtsZ. Right? So you really need to use the fragments. And others have recently converged on exactly the same idea. But there's a lot of benefits to modeling even full-length protein-protein interactions using fragments. We also got into then this question of how strong is the binding and can we get a sense of that? And so we don't currently have the kinds of systematic experimental data that we'd love to add to the mix on the binding strength of these fragments. But what we do have, of course, is modeling. And so we've used Rosetta to predict the extent of binding, right, using the Rosetta binding energy for each of our fragments from these various proteins. And we compared that to the same measurement for known peptide binders of proteins that are in the PepPro database. And what we saw is that the binding energies in Rosetta were very similar between our fragments and known structurally characterized peptide binders. They're only about 20% weaker when we constrain to allow essentially only side chain rearrangements in these energy models. But then if we actually allow relaxation of the default settings, we also see the gap reduces further, right? And when we look at what happens upon relaxation, what we basically see is that we don't see any substantial rearrangement of the predicted binding. So this is nice because this suggests that really you can use the FragFold or AlphaFold model as a really decent model without having to do this further optimization. Of course, you can probably improve the model, but we don't see a huge effect. I'm just showing a few examples here where we have in green the FragFold model, then the constrained model from Rosetta, and then the relaxed model. And basically, the main take-home message is that we see very little rearrangement. We even see that things like hydrogen bonding networks for the fragment binding as a beta strand to a beta sheet basically look the same whether or not you relax. All right. So how much time do I have, Chris? We're good.
You got questions, so.
Okay.
You got that, you got that.
Sounds good. So we also saw, we were also, of course, very interested in the specificity of these FragFold predictions. So I'm showing you a few examples of how we've looked into that, just to give you an idea. We looked at, for example, fragments tiling across FtsZ binding to FtsZ, as I showed you, which is here in the top left in this blue color. And then we looked at, for example, FtsZ fragments binding to GFP, which shouldn't be in the cell at all. And we see, for example, absolutely no binding predicted for FtsZ fragments to GFP. And similarly, no binding, if you look here on the bottom left here, no binding predicted for fragments of DNA gyrase to FtsZ. Now, sometimes you do see non-cognate binding predicted. So for example, if you look at the predicted binding of FtsZ fragments to gyrase, which is sort of the inverse of what I just showed you, then here in the bottom right, you can see that in gray, there is some predicted binding. But what's interesting is that the peaks tend to be qualitatively different. They tend to be narrower and weaker. Right. And so that's something we see pretty much across the board. And so to give you a few more examples, just to show you that we kind of really went into this, we looked at a lot of sort of not all by all, but sort of several proteins by many proteins comparison and looked at the predictions of tiling fragments across the whole protein against many potential binders. And we generally see good specificity, right, where the known or expected native binder gives the strongest and widest peaks. And we actually incorporated this peak width criterion as one of the filters for getting a higher true positive rate. Now, one exception that we can talk more about later if people are interested is we saw more off-target predictions for RPLL, which I mentioned earlier. So I think this is kind of enticing, and I'm inclined to think of this not necessarily as just a failure, but actually potentially some evidence that some interesting things are going on with RPLL, because that was the fragments that I mentioned to you previously, where we actually saw really off the charts inhibitory effects compared to the concentration of the ribosomal protein, the full-length protein, suggesting that maybe one explanation could be that there's also pleiotropic binding to other targets. So overall, we developed, I won't get into the details too much, but we developed metrics for specificity based on the density of peaks per fragment and per amino acid of the target. And looking at this peak density as a metric and the ratio of on-target to off-target peak density as a measure of specificity, we saw generally good specificity of about 10-fold, but it does vary protein to protein, with RPLL being the standout example from what we've looked at. All right, so to come full circle, we then returned to our experiments and did deep mutational scanning of these protein fragments, right? And we looked at a series of fragments tiling multiple fragments across a series of interfaces from diverse proteins. So I'm showing you here the mutational sensitivity from these experiments on average for each site, where we have characteristic mutations to different types of residues at each of the 30 positions of each fragment. So in red, you see shading in red corresponding to sites where mutating the fragment mutating that residue tends to decrease inhibitory activity, so you get a loss of function. And then in blue, we have sites where mutating that residue on average tends to increase inhibition. So one of the first things you notice is that this is a really rich landscape of mutational effects, right? And this is something I alluded to earlier. There's quite a bit of sensitivity in general of the to mutation of these fragments. And I think this is actually very consistent with the idea that these are marginally stable folders that are doing a lot of folding and unbinding. And so there's a lot of ways you can modulate the folding. And that's borne out by how these sites correlate with sites that are involved in folding. But we also see that a lot of these sites really are mapping to exactly the predicted interfaces. And in particular, a much larger fraction of the contacts are sensitive than residues overall. And that was highly statistically significant. And so, yeah, go ahead.
How do you look for proteins which have conformational changes? How do you look? Do you look at the two conformations, or do you look at something in between also?
Okay, great question. So the question is, how do we look at proteins with conformational changes, and do we look at one conformation or the other, or something in between? We haven't done a whole lot of that. I think it's a really great direction. But yeah, we have started to get into that, and I think the approach we've been following currently is that we will look at as many versions of the target as we can, right? If we have any information on that, because as we've seen and others have seen, changing things like say the fragment length or changing which portion of a target you expose for predictions can change the result quite a bit, right? And so yeah, I think that'll be very important for things like fold switches or multi-state proteins. So overall, we see that there's actually a really strong preponderance of these fragments at modeled contact sites that are highly mutationally sensitive. Yeah.
So I'm actually really intrigued by out of this kind of looks like the areas that are red are directly maybe hotspotted blue word target, and the blue areas are a little bit. I'm wondering if you have any as to why the blue areas are better. But on ground contact
Yeah, it's a great question. And we'll get into this a bit on the next slide, but basically the question is, yes, to what extent is there, it seems like there's a correlation where red sites that are loss of function sites that have decreased inhibition when you mutate them are more likely to be at interfaces, but not so much for blue sites. And that's borne out statistically, actually, what you're saying is true. And so I think the simplest explanation, and one that when we look more deeply at specific examples seems to be the case, is that a lot of these blue sites are actually affecting folding of the fragment, not directly binding. But of course, that all affects function, right? And we'll look at a few examples. There are exceptions, but that does seem to be the trend. And I think that actually makes a good amount of sense. So overall, we see that we're able to really see that these models that we make with FragFold are capturing, we think, key features of the binding and of the function of these protein fragments in cells, right? And I'll note that this actually even holds up in the case of these novel interactions we predict, which had no previously known structure. So we see, for example, that for FtsZ binding, in this case, to FtsZ itself with the C-terminal fragment, we see this really strong correlation where most of these red sites are at the interface. Of course, there are other features here as well, and we're equally interested in why other residues may also have important functions, perhaps even by binding other proteins. And so digging into the mechanistic details a little further, we could actually look at to what extent we can rationalize the loss of function or gain of function mechanisms of specific mutations rather than the average effects at every position when we mutate the residue. And we looked at, and these examples I'll show you, I'm showing you some of the strongest examples both for loss of function and gain of function in the entire library. So for instance, we see a number of cases where there's some, say, hydrophobic residue that's involved in either a key folding interaction or a key binding interaction. I have one example of each shown here, right? And when you mutate this, say, valine 171 from this FtsZ fragment to aspartate or lysine, that's very bad, and presumably because you're really interfering with the ability of this fragment to actually fold and bind in the first place. Whereas mutations to hydrophobic residues are fine, right? And we see something very similar for this leucine 178 from this fragment, which is directly involved in the modeled interaction. The effects are, again, very consistent with the model, where you can change it to a phenylalanine. Even that's not great, though. And it gets worse and worse, especially if you get into charged amino acids, right? So this seems to be a case where the details of how the residues interlock are also really important. But what was, I think, even more exciting and somewhat unexpected for us was actually that we can also uncover superior inhibitory fragments in really high throughput. So we can systematically find better versions of these peptides with this assay we've developed. And sometimes they're a lot better. And so one example, which is one of the strongest cases we see, is shown here in purple on the right. And what we see is that this valine 262 in this FtsZ fragment is sort of right at the base of a hairpin-like structure formed by the fragment. And so it looks like a folding effect, right? Where you, if you mutate this residue to, say, a phenylalanine or a leucine, you can actually make a much better fragment, probably by making it fold much better and then not have to fold as much upon binding. And the opposite holds as well, right? You make it an alanine or a charged residue, you have a lot of problems. Okay, so in conclusion, I've showed you today how protein fragments are generalizable, genetically encodable inhibitors against any protein. I've showed you how our computational fragment scanning approach, which we've called FragFold, allows for high confidence prediction of these inhibitory fragments across proteins with diverse structural and functional features. And I've showed you how FragFold actually allows us to predict novel, previously unknown binding modes that agree with experimental data. I've also showed you how these high throughput computational and experimental approaches are actually highly complementary for both discovering novel peptide inhibitors and interpreting their mechanisms of action in cells. So looking forward, I'm really excited, as Chris alluded to have been awarded the K99 award to further pursue this work, both as I wrap up my postdoc and begin my independent position. And so I'm really excited to leverage these scalable tools I've developed to develop a real systems biology of protein fragments in living cells. And in particular, really excited to then use these protein fragments as generalizable probes to study protein structure-function relationships in their natural environment and as a function of biological conditions. And also to dissect in a really systematic way the physiological roles and the evolutionary implications of protein fragments that can arise in cells naturally. And of course, we're also extremely excited about these novel kinds of protein fragments as new kinds of therapeutic peptides, with a lot of potential applications. And with our approaches, we think a really quite rapid way of both predicting and measuring their effects in cells. So with that, I want to thank Stan and Jean for all their mentorship and support. Thank you to everyone in both labs. And a special thank you to Sebastian and Amy, with whom it's just been a real joy to work on these computational approaches. Thanks also to my funding and you for your attention, and I'd be happy to take any questions. First question, yes, please.
Do you see ways to extend this to ways to predict the effects of cyclizing the peptides,
Yeah, that's something we're really excited about. So Sergey and others have recently developed approaches to directly sort of cyclize in AlphaFold predictions, and we're exactly hoping to plug that in and basically make this exact same maps we've made now with cyclic peptides and see which ones still maintain function based on our predictions, and then we could go after those experimentally. So great point. Yeah, Chris.
Super exciting work. I couldn't help but notice your final slide saying make inhibitors for any protein. Seems like the ones you showed today are all required for life. And so just wondering about your thoughts on how you'll extend this to proteins that are not directly required for viability.
Yeah, so that's a great point. We think the idea of making these fragments as generalizable binders is pretty universalizable. However, our assay, as everything I've shown you so far in our assay, leverage the idea there are a lot of proteins that are just important for growth. They don't have to be essential, but important for growth under some condition. And that lets us do this in really high throughput experimentally. So I think we can separate what we can easily assay experimentally with what we can do with our prediction approaches, right? One thing we're, of course, excited about, though, is expanding further across the proteome, which we're already working on, right? And then leveraging growth under many different conditions, sort of get a battery of conditions in a sort of multidimensional space of inhibition as a function of different conditions where different proteins become important. And we're seeing lots of evidence that you can see that, right? That it really depends on what the cell's experiencing. So I think we can build up to do this experimentally on many, many proteins. And we can predict the binders, we think, for almost any protein. Yeah. Yeah, please go ahead.
Great. I have two questions. One is technical, the other one's biological. The technical one is, how sort of reliant is FragFold on the MSA representation of the peptide? Do you have to have an MSA for this small fragment? And if you were just to fold the fragment by itself, with or without an MSA, an AlphaFold, do you get the structure of that fragment predicted as if it were part of the protein that it came from, or is it on average different? That's my first, that's a technical question.
Okay, well, let's do that one first. So yeah, great question. We've done a little bit of work on this. I think there's a lot, it's an exciting question and there's more to do. But preliminarily, we've looked at what if you give no MSA for the fragments? And what we see is that most of the time, the fragments sort of fail to fold in the prediction and then therefore also fail to bind. But in the cases where they succeed, they basically universally have succeeded in folding to a native-like configuration, even without the MSA. So the way we see it, right, I think the MSA is a shortcut to predicting a folded structure, and it biases it towards a folded structure similar to what you'd see with folding and binding. What we're really predicting is the binding of the fragment to the target. We're not really predicting the folding, and that the MSA kind of gives us a way to push it towards that native-like conformation. And so a big question mark with our predictions is, the way we're doing this, we don't know how strong the affinity is. We can't straightforwardly map this affinity because we don't know how much entropic cost there is to folding, right? And the way we're doing this, FragFold certainly doesn't know that. But I think it would be cool to develop further in that direction in terms of actually modeling that, right? Yep.
The other question I had was, can you just talk a little, I'm very interested in this idea of, the protein sequences, these little fragments sort of build up biologically through proteolysis or through truncated expression at the ribosome. Do you see, I just want to learn a little bit more about that. So do you see in biology that it's like the N-terminus, you get N-terminal truncations, you get C-terminal truncations, and that these are where, you sort of are saying that any fragment in a protein sequence is sort of susceptible to just being a fragment on its own inside of a cell. And I'm wondering, are there rules to that in what you might find as a fragment? And do you see that in like evolution or something to suppress that?
Yeah. Well, that's a great question. It's actually, you've pointed to exactly one of the major thrusts I'm pursuing in my own future lab. That I think there's a lot there, and I'm really excited about it, right? Basically, of course, it's not totally random. And one of the things we're very interested in are actually currently working on is libraries of fragments that are then, instead of just being systematic libraries of all fragments, biased towards the kinds of fragments that arise in cells naturally. And yes, there are definitely principles. For example, only certain kinds of fragments are likely to arise from proteolysis in a given cell type. And so that's something we're very interested in. And so, right, there's, but, you know, when it comes to errors in gene expression, you can, in principle, get, you know, proteins that truncate anywhere. And so I think it really depends on the mechanism. And we're kind of interested in all of the mechanisms, right? But it's totally not unbiased in general. And we're particularly interested, of course, in the kinds of fragments that are likely to arise at higher levels, or are fragments of proteins that are themselves at a low level, where you'd be much more likely than to see an interesting function. Right? Yeah.
I also have a question about the assay model you talked about. So you are using rather short sequences, like 30 to 50 amino acids. And in my knowledge, the sequences with this length usually don't find enough match from the UniRAP database. Is there any relationship between how many sequences you can find and the final performance of the structural prediction or performance of the model?
That's a great question. I'm sure that there is definitely a relationship, and we also see this. So we've done things both ways. But what we found that really helps is that we actually build the MSA of the full length protein and then fragment the MSA. So essentially, we align more things together, not relying on the fragment alone to be the basis of all the alignment. And I think that helps us a lot. And we see a number of cases where the prediction actually, yes, failed because they're, like, for example, the C-terminal IDR of FtsZ, the predictions basically failed to predict binding if we used only the MSA of the fragment because there just wasn't enough information. So it's exactly as you say. So that's very important. Yeah. Yes, in the back, please.
Benchmarking
Can you say which kind of databases? Ah, yes, that's also something we're very interested in. We haven't tried it yet, but I would love to talk more about that. Yes, the question, sorry, repeating the question. The question was, have we tried benchmarking or comparing the FragFold predictions against data on peptide MHC binding? Yes, we're very interested in that, but we haven't done it yet. But I think that would be a quite straightforward and interesting application, right? Yeah, absolutely.
We thought of like molecular switches and PTM binding. So thinking about like signaling pathways, we hit it specifically upon say phospholipid.
Okay, so the question is, have we thought about switches where we could think about binding specifically to say a phosphorylated form of a protein, for example, right? Great question. We have so far been doing everything with AlphaFold 2, not AlphaFold 3. I think we would love to build further in the direction of directly actually incorporating modifications. We could try something like phosphomimetics, but also AlphaFold's not necessarily that sensitive to single amino acid changes. So yeah, I would love to. I think AlphaFold 3 would probably be the way to go. And I guess now we have the code, so we should do that. Yeah, thanks. Yeah.
Have you ever seen like those fragments actually bind protein, but actually help to survive in the cell?
Yeah, the
like,
question is
control protein quality control, like something like.
Yeah, the question is, do we ever see fragments that bind and appear to actually help in terms of, say, in our readout, maybe increase growth? Great question. Actually, something I'm also very excited to go much further into in the future. And yes, we see that they're rare. And we're very interested, right? It's a lot easier, I think, in general, as you might imagine, I think, to break an interaction than to have an interaction be helpful, especially when you're just using native fragments. But we do see it. And the cases we see are strong and reproducible. And so we're very interested in how those are working. And of course, how they might be, useful. Yeah. Yeah, please.
I have a technical question. When you co-fold your protein with the peptide, and you're not using multi-marker, right? So you're using AlphaFold to predict the folding of a single polypeptide chain. Am I correct? Now you're just concatenating the alignment.
No, it's not a single chain. Yeah. So initially, I think Sergey and others developed this trick. I don't want to misattribute this. A few people, I think, came up with a trick of using a long glycine linker, and that was how AlphaFold 2 was originally used for binding interactions. But then I think Mingyoung Baek figured out that you could actually use a residue index gap And so when you make a big enough residue index gap in the input, then actually the model starts treating them as separate chains, which can be at any distance, and there's no invisible chain between them.
I see. So this
Yep.
must come from predicting structures in PDB or gaps or just or region or result.
Well, this kind of gap I'm talking about is more an abstract gap, like in the representations.
No, I understand, but
yeah.
it still thinks of it as a contiguous chain, right? That's where I was.
No, well, I think it's essentially modeling them as separate chains, as far as I understand it, the input to
No,
call
what's
out.
the difference with-
Well, I think I'm not sure about the low level representation in multiple, but I think one of the major differences is that it was actually trained directly on multi-chain data in the PDB, and so I think the representation there explicitly had multiple chains rather than a large residue index gap. But it sounds like the outcome is similar in terms of a large enough gap. It sounds like I don't want to mischaracterize this, but I get the sense that the model kind of gives up on having any distance constraint with a large enough index gap, right? Yeah. Yeah, Chris.
So there's some questions from the folks online.
Great.
Min Chen says, great talk. Could you clarify the specific scale of your experimental screenings when you refer to them as high throughput for each target protein?
Great question. Yes. So the question is, yeah, can I talk more about what scale we actually did these experiments on? Right. So for each protein, we had every possible fragment of a given length and several lengths. So it's roughly on the order of the number of fragments of the length of that protein multiplied by however many fragment lengths you're interested in. So for example, for FtsZ, we're talking about 354 fragments at 30 amino acids and similar number at 50 and 20 amino acids. All told across these 10 or so diverse proteins, and what we've published with this work so far, what we see is that we had about 12,000 fragments in the library. But that's not a fundamental limitation of the method at all. It's just a matter of how much you want to transform into cells, how big of a library you want to order of oligos, say from Twist, and how much money you want to spend on DNA sequencing, right? But it's not really a fundamental limitation in terms of the scale of how we can measure these effects in high throughput. And we're going much larger now to proteome scale.
And one more from Richard Phillips. Great work. You mentioned the fragment size was an important predictor of successful inhibition. What was the average effective fragment size?
Okay, so the question is, yeah, what was the average effective fragment size, and did we see a trend there? Yeah, basically, I would say under these conditions, we saw that 30 amino acid fragments were overall the best, and we didn't really see individual proteins that deviated from that. Like that trend held across diverse proteins. Yeah.
A follow-up on that question, is that something that comes out of the way that you compute it, or do you think that there's a physical explanation for that?
Okay, so the question is, does that come out of the way we compute it, or is there a physical explanation? I think when I responded to the previous question, I was referring only to our experimental work. In terms of the ability of FragFold using AlphaFold to predict these interactions, we haven't characterized it at the same scale as we have measured it. So we haven't actually systematically looked at fragment length effects, but preliminarily, my sense is that FragFold and AlphaFold are not really nearly as sensitive to fragment lengths as reality. And so I'm not sure whether that's a detail that's faithfully captured enough due to the various factors at play, probably because of the bias towards folding, even with a really short fragment. But that's something we'd like to explore more. I would say it remains to be explored. We haven't really done that systematically. Yeah, on the computational side. Yeah, go ahead.
So, just thinking, this sort of reminds me of some work that I don't know how long ago it was, of sort of looking at heavy chain CDR3 as fragments, doing a peptide array of CDR3 loops against, and to see if it would still bind the same antigen that the Fab or the antibody bound.
Like a use display kind of.
I think it was like more of a peptide array on the
okay.
slide.
Yeah.
And they did find that actually a large number of these CDR loops could still bind their proteins. And I'm just wondering if I guess with FragFold, you wouldn't have the MSA really to rely on folding the CDR3 loop. But I wonder if you've tried it, or if maybe you should try it. Maybe you could use a template or something. I don't know how to do it, but there might be, I don't know if it would be interesting in terms of modeling CDR.
Yeah, I think it's a great idea. So no, we haven't tried it, but I think I'd love to. And you're right. Yeah, the MSA is sort of a challenge, but we should think about other ways to get around the MSA. Absolutely. Yeah. They're in the back.
I'm wondering if when you selected your targets, were you looking for cases where it was like likely a linear epitope, basically, that's doing the interaction, or was it just unselected, looking at proteins required for viability?
Question is whether we were looking, when we selected our targets, whether I was looking for linear epitopes. No, not in any way. I was picking these proteins purely on structural and functional features. I wanted proteins that were involved in diverse processes across central dogma and cell division and so forth. And so I wasn't thinking about that at all. And so I think one of the cool surprises or results from the work was that fragments seem to be able to do this pretty generalizably, whether or not you would traditionally think of this as a linear epitope. Of course, there are linear epitope-like features, though, in each of these interactions. If you think about a loop or sometimes a helix, which there's just 5 or 10 amino acids, we do kind of see that, right? But we didn't select, we just kind of picked targets that were diverse and then looked for inhibitory effects systematically. Yep. Yeah, please go ahead.
For the optimal fragment demands, instead of counting them, did you try to expand from the hotspot and see where is the optimal length, where you have your hotspot that's in direction phase, you add one amino acid to the left and right, and gradually expand using fragment fold to test and see where would be the optimal and how.
Okay, so the question is, yeah, have we tried kind of building out, or maybe in, I guess, starting from a fragment that works, how much of it do we really need from a given interaction, say, right? And so, yeah, we haven't done that, but when we look at the 20, 30, and 50 amino acid fragments, we see something interesting, and you can see this in the 2022 paper, where when we look at, say, this interaction site 1 from FtsZ, the shorter the fragment is, right, the more it's actually mapping just sort of the something close to the tip where this actual interaction is happening, with the tip of this helix binding to this loop on the other side of the interface. So we kind of see that as you get shorter and shorter fragments, you can map more and more finely. But there's this trade-off we see where you're mapping more finely, but the effect size of inhibition is getting weaker and weaker, presumably because you can't bind as effectively or fold as effectively and therefore bind as effectively. Yeah. So, but yeah, great idea to like really get it down to residue resolution. We would love to do that. Yeah.
All right. Well, let's thank Andrew one more time for an awesome talk tonight. Yeah. Fantastic. So there's still a little bit of beer and quite a bit of pizza. So please hang out, chat with our speaker some more, and we'll see you in December for our last meeting of 2024 back across the Charles at MIT, where Maria Sola Colom is going to share an awesome story with us. So yeah, have a good night. Thanks again.
Thank you.